So once more, welcome everyone to today's webinar, Data Management and Research Integrity, which is best practice transfer from the University of Milan within the OIT Boost project. My name is Ursula and I am the assistant of this in this project. And before we move to the real content of the webinar, just a few words about what is One Health Boost, One OH Boost. So this is 3 years initiative happening thanks to funding from European Union from Horizon program. And this initiative, this project aims to develop research and innovation excellence and as well to improve the management capacities of Estonian University of Life Sciences and Tartu University in the area of One Health. And this is happening through the partnership with the University of Helsinki and University of Milan. And representatives of this university, we are happy to host today. And you will have the opportunity to learn from the experts of the data management and research integrity topic. So we have associate professors of animal anatomy and physiology, which is Professor Valentina Lodde, which introduced you the topic of image integrity in scientific publication, and also Associate Professor Federica Franciosi, who will introduce the topic of conducting and reporting quantitative real-time PCR experiments according to the MIQE Guidelines. So these are our wonderful speakers. If you have any questions to their presentation, to the knowledge case studies that they will be sharing, please raise your hand by using the Zoom button or write your question in the chat. As you see, the webinar is being recorded and later will be shared on our OH Boost YouTube channel. That's everything from my side. So Valentina and Federica, the floor is yours. Thank you.
Thank you very much for the presentation and welcome everyone. Yeah, just a few words before before starting with the presentation. I really would like to thank the project and Professor Tiziana Brevini for including us in this initiative. So we will try to bring our what we know about this subject to something that we work with every day. So just a quick question for the audience. Can you let us know how, are you PhD students, professors, researchers? Are you involved in life science rather than other, just to give us an idea of your background so that we can, focus our examples a little bit better if you can. Okay, so I will pretend that you are mostly, let's say, PhD students or master students, but feel free anytime to, so we keep, I actually kept the presentation a little bit basic, but if you think that it's too basic for you, just raise your hand and feel really very free to ask a more specific question if this is the case. So really feel free to the more interactive, the better. So feel free to do that. Okay, so I'm gonna share my screen. And now you should be able to see the first slide, right?
Yes.
Okay. So this is the title of today's topic. That's my name. And me and Federica, we work at the Reproductive and Developmental Biology Laboratory of the University of Milan at the Department of Veterinary and Animal Science. We work with in the topic of reproduction, oocyte, embryo. So most of the things that we are going to show are on that model. But again, feel free to ask things if this is not your topic, because the issue of image integrity related to scientific publication applies to every field in research. Everything that we want to publish most of the time comes with images. It's not only about text and data or graphs and tables, but if you look around, mostly in life science, in any paper there is an image that it's there to show as an example of representative images, or that want to show example of part of the work that has been conducted and has been published. So the issue of image integrity when it comes to publication is becoming very hot, I would say. And yesterday, when I was preparing the slides for this presentation, I very quickly Googled again image integrity and scientific publication, and you have a very, very long list of articles and papers saying how much the scientific community should be aware of what it is and how to avoid it, because this is an issue that can really affect the way in which we as scientists are seen by people. And if the the public start to question about what we publish and what we show, they're immediately going to argue that what we say is not the truth, and they will be starting not trusting us anymore. And we don't really want that. I mean, we saw all the mess that happened with COVID and with people not trusting scientists and us being in the spot because they were skeptical that yeah, scientists say something, but it's it's hard to let them trust what we have to say. It's really hard. And the way we have to behave, it's being as more transparent as we can in everything we do. And images, it's an issue because it's very easy to manipulate images, even though we are in good faith. I don't know how to say. It's very easy to make mistakes when it comes to images and preparing images for the publication, to introduce something that it wasn't there in the first place. And when it goes in the publication and an issue comes up, then the fault is on the scientist that published those things. But most of the time, the scientist is not aware of what the issue is with their images. So there is a huge interest for the scientific community as a whole to be aware of this and to learn how we can avoid it. And that's why every year at our PhD course, we do a course about how to handle images, and we talk about a lot about these topics. So again, here I just put a couple of references. So if you click and want to read, this is a very nice overview. It's a little scary, at least to me, because just reading the title, it says that more than 10,000 research papers were retracted in 2023, which is a huge number. If you read the and if a paper is retracted, it's because sorry. Okay. We if you go through the through this paper, it says that of course not all of them were retracted because of image issues. but a good 30% of them were retracted because there were problems in the images. And this is a huge issue for us, and we should try to avoid it as much as we can. Just a little step before starting the real topic. I guess we are all familiar here about the editorial flow that brings to a scientific, to our data being published in a scientific journal. So everything starts, just very quickly to summarize, just in case there's someone that is not very well aware about the whole process. Everything starts from the researchers, that it's me and you, running experiments, collecting data, being in the lab or in the field, most of the time running nice things and enjoy what we do. And this is a very the time in which we acquire the images that we want to put in our publication, because what we put in our publication, this is probably the bottom line of my seminar, it's that we don't publish images just because they're pretty. We publish images because they are part of the data that we collected in that particular experiment. So what we want to, in the end, put in our scientific publication are a representative image that were acquired in that particular set of experiment that is part of the publication of the data that we want to publish. So when we run all the experiment, we're happy with the story that we want to tell. We put together the manuscript, and the manuscript is composed by several pieces, the title, the abstract, the introduction, the methods, results, and all the different parts that then we see very nicely organized in the published paper. And part of it, the way we submit we organize the manuscript, it's we write in a Word document the different pieces according to the author guidelines, and these are different based on the journal that we pick. But most of the time we have to not only write the text and the data and the materials and methods and the results, but we have to prepare figures and tables. So now we talk about figures in a specific way. So everything goes to the editor when we submit the manuscript, and the editor-in-chief is the one, or the associated editors for certain journals are the people that decide if the paper is good enough to be processed by the journal, and the editor then pick the reviewers that are expert in the specific field. So who is going to read your manuscript at first are expert in your field. They know very well the topic, or they should know very well the topic, and are able to not only judge, but give you eventually advice on what is not very clear, and they can suggest changes and stuff like that. So the reviewers can raise questions, and most of the time they have issues or something that is not very clear. So everything goes to the editor that takes the final decision, and if there's issue, the manuscript go back to the researcher that can fix the issues, or sometimes can ask for additional experiments or stuff like that. So if the scientists want to fix the issue, maybe run another experiment, and then everything go back to the editor, the reviewers, and blah, blah, blah. And this circle can take months. It's a very long process sometimes to publish some data. But to me, at least, this system, it's not perfect, but it's the best we can have now. Trusting reviewers, it's an important issue. And having people that are our peers giving us suggestion is the best way to go. At least now I don't know what else can we do. So when everything goes okay, and everybody's happy, reviewers are happy, and the editor is happy, then all the different parts goes to the publisher. The publisher, in this case, the publisher and the editorial people, the editorial staff, they don't know nothing about the science that we want, that we are giving them, that we are publishing. They just try to put all the different pieces in a nice page, in a nice way that then fit in the page for the publication, for the final publication, that in the end it's a page of a journal. Okay, so when the paper is accepted, the path is not finished yet. There is this big part that is the editorial processing, and in that case, we need to be sure that the figures that we have submitted in the first place comply with the editorial requirements. So we want two things when it comes for to understand what kind of images we need to give to the journal. One is that they tell the truth, that they are real representative image of the experiment that will run for that particular set of experiment that we want to publish. And the second is that they have to comply to the editorial policy and the editorial requirements in terms of size, resolution, and all these things. This is very tricky for a scientist because we are not, I mean, maybe probably I'm not, maybe some of you are, but I don't know. We are not, how to say, what's the word in English? I mean, we are scientists. We are not expert in image manipulation, right? So we need to know, a scientist has to know at least the basic of image adjustment to make something that it's good for publication, because sometimes we have to cut images, to enlarge images, to change the resolution and stuff like that. So we have to be aware we have to know how to do that. So probably you already know all this story. So in all this scenario, why this seminar? A seminar on image integrity that is focused specifically on images is because 50% of a scientific paper is actually in the images. Because when reviewers read in the first place your study, your research, one of the first things that they are going to look are the images, and we need to do that. We have to communicate science to people. If we keep our data in the lab, it's not going to work. We have to go out and let people know what we're doing in our lab and share our knowledge and our data. And we do it through, of course, scientific publication. And again, 50% of, if we want to communicate in a in a powerful way, let's say, our images have to be good, okay, have to tell something. The reviewer should understand the flow of the work and what we have to say. Even before reading, they should be able to look at the different images and the different figures, and they should be able to understand the story. Then they will go in more detail, read the text, but the first impression is the images. So if we convince the reviewers and our colleagues that our science is good through the images, they will read the rest of the paper with a different mood. I mean, this is at least what is my experience. Always remember that high quality images and figures reflect most of the time the high quality of the scientific lab and the institution. When you publish something, it's not just about you. It's not just about you as a person or you and your supervisor. The name of your institution, it's there. So if you do something that is wrong, they're not going to blame on you, but they're going to, say that the institution as a whole has problems. So, do not underestimate this responsibility, because I think this is sometimes it happens and I say, yeah, I have to wrap up. I'm finishing my PhD. I just want to publish. So who cares? I'm going to move on in a new place. But this is not the way to go. So we need to be sure that the things that we have to say are good, because we are representing our lab and our institution. Okay. So when we publish something, just to sum up, when we publish a scientific paper that include images, what we want is to prevent rejection. This is kind of important to speed up the process, and not only prevent rejection from the reviewers that are going to argue on the specific data, the experimental design and stuff like that, but also from the publisher that might say, the figure that you have just submitted doesn't comply with the editorial requirements. They can say they are too small, they're too big, they are not enough quality, they are not the right size. And this is enough for them to see, to give it back to you immediately and start all over again. Sometimes if you do not prepare the figure in the right format with the right resolution and stuff, you're not even going to be able to submit the paper, because you have to upload the figure in the system. If the figure is not the right size, you're not going to be able to submit the paper. So you have to know how to do. So there's technical issue and more scientific issue. And most importantly, and I think this is the part in which I want to focus the most, is that we want to avoid unaware, and I want to underline the unaware word, misconduct, because sometimes we can have some comments from people that says, these images of yours have this problem and this problem and this problem. So what did you do? And they're gonna argue on that. And you maybe, you did nothing. You are really in good faith. I mean, you are sure that you didn't do anything wrong purposely, but maybe you introduced some changes in a way that you don't know and you cannot control. And this most of the time happens using all the programs that are meant to, organize the images, cropping images, adjusting the contrast, the colors and stuff like that. So our enemy are the programs that we use for image analysis, not image analysis, sorry, to prepare the figures. That most of the time it's Photoshop, this kind of programs that are not meant to handle scientific images. Those programs are meant to handle nice pictures of photographers and stuff like that. So they have all the tools to introduce changes that are, of course, allowed and required when we want to show, I don't know, art or stuff like that, but cannot be done when it comes to scientific publication. Okay, so all the tools that are in Photoshop, we can use 1% of those when we use Photoshop to prepare images. So we have to be aware of that. Okay, so my task in this last 40 minutes, I guess, is to focus on explain what is a scientific figure and what are all the steps that lead to unaware misconduct and what can we do to avoid it. This is my take-home message. This is what I want to, give you. Okay, so if we look inside a scientific publication, figure, it's what we read here. It's a group of images, graphs, gel pictures, cartoons, line art, and so on that illustrate your results. Always remember, as it's written here, that everything you show has to be and must be a true representation of what you saw with your eyes, or a true representation of the mean of the data. When you deal with when you create a graph, for example, the graph, you show the mean of data that were acquired in different experiments, and you show a bar graph, something like that. You cannot change the number to make the graph different, right? You will never do that. In the same way, you don't have to modify the image to change the information that are in that image, right? Because images are nothing else than data. Everything when we acquire images, we are acquiring data. Sometimes we use those images to do, I don't know, fluorescence intensity analysis, or when we have gel pictures, we use those images to calculate the density of the bands and stuff like that. So we really have to treat those images as data. We don't want to introduce any changes to what we saw when we were acquiring the images. Also, this is something that I want to highlight. The image that you publish in a paper has to be part of the set of experiments that were run for that set of data. If, for example, you have the images and say, no, this is not very good, we have this background, it's not very nice. Yeah, my colleague in the same lab did this experiment, or I did this experiment for another set of experiments for another project, and that one was much prettier and nicer. And maybe someone can be tempted to use that other image because it's just prettier. This is not going to work. I mean, this is not the way to go. You should use and you should show a representative image of that set of experiment that you're going to publish. Okay. Of course, this is different if you want to write a review or stuff like that. Of course, that in that case is different. So the images have to be informative and not just pretty. Okay, this is a pretty picture are for the first page. It's not for the core of your publication. And so they have to be informative and must comply with the editorial guidelines, as we were saying. They have to be of a certain size, so they have to fit in a page, and we will be talking about this in a second. They have to have a minimum resolution in terms of DPI, so dot per inch. They have to be in a specific format, and usually the required format is TIFF. And always remember that a figure is nothing without a figure capture. So, which is the text that you prepare to explain what you want to show in that particular figure. So this is an example of some two images that were part of one of our publication. This is the paper, if you want to have a look. So in this image, you see that the figure itself, it's the combination of different things. In this one, it's the combination of Western blot gels, in which we wanted to show the expression of a particular protein in a control with the antibody or in the negative control, and then the expression of these other proteins in different experimental conditions. So, and then there is a cartoon explaining the domains of the protein. So the way you arrange this is that you have to submit a figure as one file, not the different pieces, and then ask to the editor to put them together, because it's you that decide the way you want to tell the story. So you cannot give freedom to the editor. They're not going to do it anyways. They are going to ask something like that. So it's you telling the story. So it's you that want to show your pieces of the different figures in a specific way. So this is another example. If some of you it's more familiar with microscopic images, these are again the expression of different proteins, and this is the merged images, and this is the another assay counting. I don't know if you can see it, the numbers of these red dots here, and this graph. There is the counting of these different dots expressed in a numerical way in a bar graph. So the way I prepared these images were to take, in this case, for example, to take the image in the DAPI, the DAPI and the green, it's the expression of this protein, and then merging. So I had to take different images and then combine them together and put them together with the graph. So I started from maybe 1, 2, 3, 4, 5, 1, 2, 3, 4, and 5 different images and a graph that then I combined together. And then here, of course, it's the figure capture in which it's explained. What I wanted to say, it's nicely explained. The figure capture should be self-explanatory. This is something that we always want to do to help the reader to understand what is in that picture. So the idea is that the reader and the reviewers don't have to go back to the text to understand the general picture. Of course, they have to go back to understand the little details, but the overview has to be explained in the figure capture. Okay. Okay, so the way I did these images was to first acquire the images with the microscope, and then high resolution, high everything, and then combine them together using Photoshop. And then using Photoshop, I added all this other information, the lettering, what it is, the graph, etc. And these are all the writing here that are more vectorial part of the image are meant to help the reader to understand what they are looking at. Okay, so not only I had to combine them, but I had to think that this image here is meant to fit in a page. Okay, and the page has specific dimension. This is usually in European journals, they use A4 format that have 25 centimeter by around 30 centimeter. So we have to think about something that can fit in that page. So this is the first page of the paper, but if you look into the other page, I had to prepare the image in order it to fit in this space. I cannot give something that is 50 by 70 centimeter by centimeter, because otherwise the editor will have to squeeze everything in a way that I cannot control anymore. Okay. And I cannot give something that it's 2 centimeter by 3 centimeter, because that is going to be too small. Okay. So I have to go in the editorial guidelines to see what are the allowed dimension, and be strict and prepare a figure that can fit in that page. So usually what you want to look at, it's the printable size of the journal. And usually if you want to publish a figure in a column that would fill one column, you should consider around 9 centimeter width by whatever 20-30 by length. And if you want to fit 2 columns, it's around 18 centimeters. So this is the general rule. Okay, so how do I do that? How do I prepare my images in a way that I'm pretty much sure that the final result is going to be good for the publication? So here are just a few points. So the first step is, of course, that I run the experiment, and my sample, the sample that I want to use, have to be good. The sample must be perfect. I cannot have good images if my, let's say, I don't know if you are familiar with microscopic images. I cannot pretend to have a good image if my slide it's dirty, or has a high background, or there's no enough cells in the area that I want to photograph, or stuff like that. So my sample has to be good in the first place. It has to be something that I would use to acquire data, okay? And therefore, I always have to include controls, and all the settings of the photographic images have to be first settled. The acquisition parameters have to be settled on the negative control or the positive controls if you have it, and then with the same settings in which I'm sure that I don't have any background or anything else that is noise would show up in my original image, okay, with my sample. So the preparation of the samples have to be very good. It has to fit to follow all my experimental design, etc. And of course, if you are PhD students or master students, your supervisor will guide you in the best way to go to run the experiments, right? Then so after sample preparation, we have to go to the part in which we take the picture. And to take the picture, we have to be sure that we are using the appropriate device. So for photographic images, most of the time we have very good microscopes with very high quality cameras, stuff like that. So those are not really an issue. But I'm seeing more and more often people taking photographic images using their iPhones or their phone in general through the eyepiece, because maybe in that time, I don't know, maybe the camera is broken or something like that happened. So you just very quickly took the image. That it's not the way to go. I mean, it's good for your screenshot, but it's not data. It's not scientific data. This happened very frequently with gels, DNA gels or other kind of easier-ish images that do not require the microscope to look at. So be careful. Always use the proper acquisition, the appropriate device. This is not because the cameras of our phones are not high quality enough. I mean, in terms of pixels and stuff like that, they can make very high resolution images, but this is not the problem. The problem with the phone or other device is that most of the time they do images in the auto mode. So they change the image in a way that I cannot control. They use filters, they use things to make images nice, because They are meant to do other kind of images. And I cannot control. What I want to do when I run an experiment is that I want to acquire my images with the same settings. And most of the time, I cannot control the settings with phones. Okay, this is just to give you an example. The same would apply with low quality cameras. Okay, so be sure about that. Then it comes the part of image processing, in which from my original image, I have to adjust and put together with other images to prepare the image for my journal. Also in this case, you want to use the appropriate program. So the best way to go is to use these programs that you see here: Photoshop, GIMP, there is the free version of Photoshop, ImageJ, and these kind of programs that are meant to work with images, okay, and to handle pixels. You really should avoid to use PowerPoint, Word, these kind of programs that are not meant to handle images in the first place, okay? And then after we put everything together, we have to conform images and figures to the specific output. So is it a different things if I want to prepare a figure for a poster presentation in a conference, for a PowerPoint presentation, or for a scientific journal? What I have to do, it's different, okay? So you have to be aware of the, let's say, the destination of your image, okay? Okay, so the first part, the image preparation and image acquisition, is actually part of your experiment and data collection. So when you write a project, when you think about your experiment, you very well decide which protocol to use, and so you're gonna think very in a detailed way, the way you're gonna write your experiment and the way you are going to collect data. Nowadays, one of the things that is more and more required is to write a data management plan, in which this part has to be included. I don't know if you ever have to write a project, a PhD project or whatever. Now every funding agency, they require from the first time to put down a data management plan, in which you basically have to describe how you're going to collect the data, how you're going to name all the, now we talk about images, of course, how you are going to name the image in a way that they are traceable every minute of your process from the very beginning to the very end, where you're going to store the images, in which format, how often you're going to do backups of your device. So this is the imaging, I mean, dealing with images when they are data in a data management plan is a big chunk of the data management plan, okay, especially for some projects. Then it comes to the image processing, and then part of the conforming images and figures to specific output. And this is where there is a high risk of potential potential alteration of your data, okay? And if there is alteration of your visual data, then your images as data can be misinterpreted, okay? And this can be done purposely, and do not underestimate that all around the world, there's people that want to show something that it wasn't there in the first place. This, I mean, we have to accept that as in normal life, that good people, there's good people and bad people. And I think the scientific community doesn't make any exception. So of course, it's a very, very, very minority, but we want to exclude those people. And so we have to have the tools to understand if someone is doing wrong things. Because if they do something wrong, then as a whole, the scientific community would be, responsible for that. It's our responsibility to control our community. It's like, who pay taxes, who doesn't. It's the same. If there's a very, very small amount of people that do not want to pay taxes and do not pay taxes, and then it's the whole community that it's not happy about that. Okay, here is the same. Maybe there is one people on a million, but that one people we want to tell that you cannot do that because you are ruining our work. Okay. And so we have to have the tools to recognize this when we work as reviewers, when we read papers. If we spot something, it's our responsibility to speak up. I mean, this is the way I feel. I'm sorry if I'm a little too harsh, maybe, I don't know, but I think this is something that has to be said. Okay. Okay, so, but I'm not here, of course, to say that you cannot do bad things. I'm here to say that you have to know the tools to avoid doing bad things in a way that you don't know. Okay, so how do we do that? We just have to follow the rules. It's very simple. Just follow the rules. Okay. And where do we find the rules? The rules are here. So first of all, we have to be aware that when it comes to images, there's two types of images. The one that we acquire that are meant to measure optical density. The examples that we did before, when we run a gel, a Western blot gel, sometimes we have to do image analysis to measure the optical density of the band, or when we run immunohistochemistry, immunofluorescence, or stuff like that, we want to use specific programs to measure the amount of the fluorescence intensity of a particular cell or whatever, that it's proportional of the amount of protein that it's expressed in that particular area of the cell. So the images that I have to use with this kind of program, and most of the time is ImageJ, I cannot do anything with those images. Nothing, zero. Like I have to take the picture, import with my device. I have to import it in ImageJ, measure, close it. There's nothing else I can do because every little change that I do is going to affect the numbers of each pixel. So I have a slide after that would explain this concept a little bit better. So if you want to do image analysis to measure optical density, you just take the pictures in the right format, use the import in ImageJ, do the quantification, close it, that's it. Of course, that especially now there is the need of processing images in a more complex way, and for those kind of images I would really suggest to follow specific course. I just did once a couple of months ago. If you want, I can advertise. I don't want to, but if there's someone that is interested, I did it with my PhD student just to see the level, and it's a life change. So I really encourage every of you that has to do some of these image processes to follow, not maybe that in particular, but to look around and do it and do it, because it's you know, especially now with artificial intelligence, everything is changing, and we have to face the new technology coming up. Okay, so there are other kind of images, on the other hand, that needs to be manipulated somehow, because we have to conform when it comes to publication. We have to change the size, sometimes change the background, reduce the noise, increase the contrast to prepare for publication. Okay, if we do all these things in an inappropriate way, the risk is that our image would be misinterpreted. Okay, so I can do this, we can do that, and we have to do that, because otherwise the image would not be accepted by the editor, but I have to know how. And in that case, again, I encourage everyone to follow specific courses that teach you how to handle Photoshop and stuff like program like that. Okay. So again, now that we have said what kind of images we have, one that we cannot touch and one that we can touch, how we can touch them, what can we do with them, where are the rules that I have to follow? There's two places in which I can look: professional guidelines and author guidelines. I have to say that now, especially, you know, these years, in the last 10 years, even, yeah, 10 years, journals are much aware of the problem with manipulating images. So most of them have very specific guidelines of what you can do if you want to submit in their journals. So one of the example is the Nature publishing groups. This is the link if you want to see their editorial policy. Another one is the group that the Rockefeller University Press that handle the Journal of Cell Biology and other groups of journal. So if you go in their website, and this is what you, for example, let's say that you want to publish a paper in the Journal of Cell Biology. First thing you have to do is to go online in their website and look what is their editorial policy and what you have to do to submit a paper, and click here. I don't know if you can see it. Yeah, this moved a little bit, sorry. But anyway, you want to click in figures and video guidelines, and they're gonna say all the things step by step of what you can do and what you cannot do. This kind of information are also present in other professional guidelines. For example, there's the Confines of Science editor. This is the link if you want to have a look. This is a group of scientists that at one point realized that this was an issue. And so they wrote the white paper on promoting integrity scientific journal publishing, and chapter 3 and 4 are specifically dedicated to digital images. There is the US Office of Research Integrity. I don't know if you know what these are, but in certain case, you can go to jail if you use in US. I don't know in Europe, but in US, if you do something that is very bad, you can go to jail. Don't think about only scientific publication, but let's think about something that is used in trials. Come si dice? Trials, right?
Clinical trial.
No, when you go
quando vai al
in
processo.
court.
Trial, yeah.
Trial in court, in which you want to show images as proof of the, I don't know, accusation or stuff like that. If you manipulate the images, it's a problem. You can really go to jail, okay? And then there is this is working pretty well. It's a group, the Committee on Publication Ethics. It's a group of people it's a group of journals actually that have set a general agreement, and good journals have to subscribe these rules. So these are the links. Of course, I don't have enough time, but if you want to have a look, then I can give you, all the links you want. And but if you Google it, it's very easy to find. So in this more official, let's say, guidelines, there is this definition that I think it's what we've been talking about. There's two kinds of manipulation. Again, one is inappropriate manipulation, and this is due to adjustment of images and data that violate the established guidelines, but does not affect the interpretation of the data. So this is, a little bit of adjustment of the background and stuff like that are okay, as long as they do not obscure or delete certain information that were in the image in the first place. And maybe, adjusting the contrast, sometimes I can delete entire stands from the gel. So these are things that maybe very young students used to do because they do not know what is the limit. So these are things that sometimes are handled by the journal or by the scientific community, just saying, what did you do with this image? This is probably you did a little bit too much with the contrast and stuff like that. Can you show me the original image to show what is the problem? So if you did something like that and you didn't want to, because you didn't know how to handle the program, you solve the issue in 1 second if you are able to show them the original image. Most of the journals now are requiring the original images that they submit, that they publish as the supplementary data. So this is solving most of the issues. So when you write your data management plan, again, we go back to the data management plan, you have to be sure that you have a plan to store your original image in a place that nobody will touch forever and are going to be there if in 10 years someone will ask me, what is the original image of this figure? This can happen. And this is a problem, I think, most of the PIs, because sometimes it's students acquiring images and stuff like that, and they save in their computer and with the random names, very creative names, and then they finish the PhD, they finish the master, they bring their computer with them, images are lost. So the PI in the data management plan have to be sure that everything stays in the lab in a safe place, okay, that can be tracked. So these inappropriate manipulation are handled in this very easy way, not a problem. The problem, of course, is fraudulent manipulation when I want to fix something because I want to hide something, because I don't want to tell the truth. I want to say something that it wasn't there. But this is not our case, so we're not talking about that. Okay, one of the main problem, and I'll try to speed up because I think time is running. And another problem with the images, it's duplication and plagiarism, because we are, I think, the aware that we cannot use an image to be published in our paper if it wasn't the set coming from the set of the new experiment that I run, but I cannot even use one picture of mine that was published in another publication unless I state it. And nowadays there is a huge problem of plagiarism, and there's people spotting those problems and reporting those problems. And now with artificial intelligence, I think this problem will be solved in 1 year, no more than that, because people are developing tools to compare. And I think this is a good way to use artificial intelligence, I think. So again, how to avoid misinterpretation and misconduct, just to sum up, because I don't have time to talk about all the slides that I prepared. I will just go through very quickly. We have to be aware of what a digital image is and all the critical steps that leads to misinterpretation, including sample preparation, image acquisition, image processing, and the last part of conforming images to comply with the specific output. Just let me show you this. This is a old but still good book. There is a list of things that you can do practically using Photoshop when it comes to scientific images. So I'm gonna give you the slide if you want. So this is just the screenshot of the book, and with the list of things that are allowed and that are not. So if you work with images, you should know what it's what is the bit depth, what is the white balance that you try to do. So in these guidelines, you can find all the limits that are, things that you can do and not. What you just give a brief, list of things that you cannot do. That can be done but need to be reported. Okay, so you can apply changes to the entire image. This can be done. You can crop and straighten the images. This can be done. You can change the mode. If you have a color image, you might want to change in grayscale or index color or RGB. You can do it as long as you know how to do it. You can change the bit depth, but sometimes, and these are in general normal things that you do, okay? So there's no need to report what you do. There's no need to write, but and these are other examples, but there are things that are not permitted at all, okay? So not permitted actions are the one listed here. Spot changes, so if you have an image, you cannot just increase the fluorescence of that one single cell. You have to apply the changes to the whole image. You cannot transfer, you cannot do copy paste of maybe one image in which you had that perfect representative cell and you want to put it in another image. So you cannot do that. You cannot do spot change, okay? And subsampling, it's something that it's really scary, but this would require another 2 hours to explain what it is. So maybe, next seminar, I don't know. Okay. So this is, again, the list of things that can be done according to the book that I just showed, but the same kind of information now are listed in the author guidelines. This is the same example that I showed you before. And if you click what I told you before, you see what you can do, what you have to do when it comes to microscopic images. So you have to declare the make and the model of the microscope, the type of magnification, all the list of information you have to give them. You have to submit images in a specific format. You have to give specific size name. You have to comply with the required size and resolution. We have to, submit images that is high resolution, and what to do when it comes to gels and stuff like that. There's a long list. And their editorial policies is this one. Now all the journals do this because with artificial intelligence it's very easy. They aware you that all accepted manuscript will go through a plagiarism and image screening check prior to publication. Images should be minimally processed and accurately reflect the original data. Our screening process examines the follow, and so this is the list of what they are going to check. So this is what I was basically telling you all this hour. So I guess that the take-home message of this webinar is why you should be worried about false images. And the answer is that it could really ruin your career and the career or supervisors and the reputation of the institution. And how to protect yourself and the institution. If you follow what it's called the RARA guidelines, you have to record everything you did. So take notes when you change images. You have to record everything you did. Acquisition setting has to be part of your notebook, okay? And you write this in the data management plan. Archive every time the originals, everything you do. And again, this is part of the data management plan. Report what you have done. If you have to change an image, just tell to the journal in the figure capture. You can tell this image was changed in this way or was cropped in this way to highlight this aspect. As long as you say it's okay, everything is fine, okay, and apply any changes globally and equally to related images. Okay, so if you want to change background to your treated group, you have to be sure that applying the same changes to the negative control would not show up any background or things that can be confused with the specific images. So just to conclude very briefly, and I'm sorry, Fede, I'm stalling some minutes from you. What are the consequences of scientific misconduct and misinterpretation? This is how really I want to, I'm gonna give just some example of some retracted papers because they're very bad and you don't want to be involved in this kind of situation. So this is the retracted paper of this particular paper. So your name is here, right? The name of your institution is here. So in the note here says that the authors retracted the October blah 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 paper cited above. Why? Because figure 1 in that paper was inappropriately reproduced from a previously published paper. In this one, this article is a duplicate of a paper that has already been published in the journal of blah, blah, blah. So they write it. And as such, this article represents a severe abuse of the scientific publishing system. This is another example. It has come to our attention that figure 3 of the title paper blah, blah, contains unacceptable levels of image manipulation and thus does not provide firm evidence of the particles reported. This is a good example. Concerns have been raised about the integrity of images represented in figure 2 and S1 of this article. This is what is the take-home message. The original blood images from which the figures were made are not available. So what this journal did is like, look, I spotted this problem. Can you give me the original so that we can double check? And they couldn't find, the scientists couldn't find the original anymore. So probably they were not organized in a way to store the images. And, you know, at the corresponding author request, the institution involved are investigating this matter. I mean, it's a big thing for you, for people that does this thing. And you have to be aware. Other examples, I mean, I can go forever because it's a huge thing. So I'm done. And of course, I will be very happy to answer questions. Just a quick thank to all the people of our lab. This is our website. If you want to contact us, free to do it. Follow us on Twitter, X, whatever it's called now, and Twitter. No, Twitter and sorry, Instagram. So we are, I'm sure also Federica will tell the same. If you want to write us, if you have more specific questions, we are very happy to collaboration. And you know, if you don't know how to use Photoshop, give me a call. We can organize something for you. And quick thank to all the people that through the years have implemented the course. These were people that participated in this seminar all over and in the course that I do for our PhD school. And so all these people gave little pieces here and there that I combined for this webinar. And thank you.
Thank you very much, Valentina, for this very engaging and insightful presentation.
Thank you.
So now it's time for the audience to ask the questions. Do we have any? Are there any questions?
I hope I didn't scare you too much. I mean, I'm a good person overall.
Can I just make a quick comment about gels, Western blots, and electrophoresis? So most of the time when we see composite images, it's like the single lane is isolated one from the other. So they should still come from the same gel. In the sense that if graphically you have something else in between and you don't want to show it because it's another experiment and you run them together, it's still okay. You can isolate the bands, but they should come from the same gel or the same film or the same membrane or things like that. So, and the example that you showed about your publication is very good because sometimes when you have gels, they are just the tiny lane that is cropped out. But if you show the full thing, you're showing that there are no extra lanes or whatever. But again, giving the original ones just a supplementary.
Yeah. I can, I can give you the slides because there's many others and there's few examples and link to papers that describes exactly what to do in the case of gels, how much you can crop, how much you can't, what is too much. Of course, I didn't, in an hour I could just give an overview, but to force you to go and look at these things. So, but I mean, I'm happy to share the slides if you want, and extra material because I can provide more.
There is a full PhD course that Valentina gives on that. So in case you're interested, it's how many? 3 ECBS?
Yes, it's organized in 2 weeks, so it's very challenging. We teach students how to use Photoshop with their images so that they don't do mistakes. Yeah. I don't know, maybe it's
Thank you. Yeah, I think Suranga has a question. Yeah.
Thank you very much, Federica. That's very interesting and very informative talk. So I just want to have a kind of a management question in terms of lab management in relation to the whole issue. So when you run your lab and when you store your data, do you really store them in digital formats or just maintain in lab books and keep it as when we were doing PhDs, we used to paste them on the pages and keep them into folders, and when we come out of the lab we have to hand over everything and then come. So what kind of advice you can give us in terms of handling this kind of
thing? The advice is to have a clear idea of what the PhD student or the student is generally doing, and don't give it for granted that they know how to handle a lab book. So instruct them very well at the beginning. So give the paper that this is the lab book. You have to report, you write everything you want, but then the minimal information that you have to write here are this, and this, because they might don't know what it's important. Okay, so what we do is that all our students have their own notebook, lab notebook, in which they write more or less what they want and their notes and stuff. They, of course, those are numbered, and at the end, when they go, we ask them to leave the paper or a scan of the lab book. But then all the information are reported in Excel files or shared files. So everything we have with our students are in shared folders. So we see what they do, basically. We go step by step and help them to organize their data in a way that then can be analyzed with the statistical program, stuff like that. For the images, that it's the most of the, maybe then Federica can give you example on omic data. I can give you example on images. Everything we do, we have a
server,
a cloud, a server, in which, so we have different microscopes with cameras, gel scanners, stuff like that. We have a code to save all the images that goes by date. So year, month, date, then there is an underscore, then there is the name, the code of the experiment, that is our own lab code, and then there is an underscore, and then there is a part for free text. So all the images have to be acquired using this first code, and then all the image of that particular experiment are organized in a folder that has the same name. So if, and then we collect all these images in the
server.
In the server, sorry. So
yeah, so it's an investment, actually.
Exactly, because you have to have the money for the server and the people that, and IT people that handle that, because it's not so this server now, it's working pretty much well. It's 15 years that we don't lose one single image. I can go back looking at the name of the experiment, the number of the experiment. 15 years ago, I have lab books. What is that? I can go back and find the original, because once it's there, then the students make a copy and work on the copy. So my original is there. Of course, it's an investment, and this server, the server, it's backupped every day. I think we have
daily. I think it is
daily.
Yes.
Yeah.
Yes. I think it's daily.
So it's part of the data management plan. You know, now it's it's. So this is my suggestion: be very clear with the students of what you want.
But not just images. If I can,
everything.
I mean,
everything.
I mean, we use the same code for all the type of experiments.
Experiments, yeah. It's easy to trace.
If they are, Excel files where you record your own observations, let's say, then they have the same type of code. Very easy to trace them back and keep them nice and clean. I think it's a little bit of a struggle at the beginning, because you have to teach every student how to do that, and maybe they don't catch it right away. But if after a year you go back and you ask things, then they realize that it's very powerful. And I think having we participated in a pilot program of the University of Milan on data management plan. It was not for all the PhD students, so it was voluntary, not mandatory, but it was super useful. And I think it's gonna get mandatory for everybody.
Yeah.
I mean, the University of Milan also have a service where they provide, I don't know how to call it, it's Dataverse, it's called, where they give a free space to students and PI to collect their own data. and store them, but we are not using it actually because there's not enough space for all the images and all the omics that we have. So we had to go and buy actually our own server.
Thank you.
So
Thank you very much.
I guess it's an institutional type of decision. If your institution can provide you with something like a cloud or something. I mean, I also use OneDrive with my students. It's working pretty good to us because with the University of Milan, this is what they are asking us to do. We were using Dropbox before, but it's we will have to pay it by our pockets because it's not compliant with University of Milan policies. So we abandoned it. Or I mean, we still use it a little bit because for some things it makes it easier. So it's really your preference and your institution. It's some sort of discussion that you should have with them.
Yes. Yeah. My main, my main advice is to let the student understand how important is that, because if they understand the importance, then they will do it. Because at the beginning they like, why I have to write all these things? It takes time. I need to be in the lab running experiment. They don't really. I mean, it's something that we have to teach. It's part of the.
Okay.
Thank you. Thank you very much. Yeah.
Thank you. Thank you very much.
Thank you.
Any other questions? I don't hear, I don't see, so I think we can now continue our learning experience and discussion. Yeah, so Federica, please, the floor is yours.
Okay, so well, thank you very much. Again, thanks, Valentina. It's always nice to hear this lecture of yours. I keep forgetting things from one year to the other, so I should be with your students again, maybe next year. So we are diving into a different problem and a possible, I mean, a proposed way to solve it in a minute. So we're going to be speaking about qPCR, quantitative PCR. And I'm not an expert in general qPCR. I'm not a molecular biologist, but I had my share deal with that, learning it and performing it since, I don't know, 15 years, more or less, I think, 10, 15 years. And my experience is mainly on reverse transcription PCR, quantitative PCR. So I don't do much DNA PCR, but mainly gene expression profiling. So maybe I'm going to be biased towards that. I'm saying it because I might be biased against that and maybe not considering the DNA part. So if you instead want to talk more about the DNA and issues and whatever experience you have, feel free to raise your hand, interrupt, speak up, open your mic. I'm totally happy with some sort of interaction. However, before we start, I have to ask if you are familiar with the technique itself. I mean, a little bit, not that you performed it on a daily basis, but that at least you know how it works. Because I'm not going to go too much into the technical details because we are going to look at it from a research integrity point of view for today. So, okay, because just in case, it's also in my presentation, but I put it on the chat. There is a link on an educational video that you might want to look at if you don't remember and need a freshen up on how qPCR works. Okay, so I think I can start my, okay, can you see my screen?
Yes, yes.
Okay, good. So we can start. Good morning again, then, everybody, and thanks for the kind introduction that Ursula gave before. Before starting, I'd like to thank the organizer and especially in the person of Professor Brevini, for the invitation to speak about the MIQE guidelines today. So as you might have guessed, we'll be talking about quantitative PCR, but not much from technical, methodological standpoint, as I've mentioned already, but rather from a research integrity point of view. And I have to apologize from the beginning for my slides being very much written. So they are not much graphical, there is a lot of words, and this is the way that I cope with talking in front of computer screen without real people in front of me. And it's not my preferred way of communicating, by the way. And I will be very basic, since I don't know the experience of the audience on the topic. But again, if I'm saying things that are too basic, just stop me and we can discuss more complex, maybe, if I can. But I'll be happy to lead the discussion, and it can be really shared. So to start, I'd ask with a show of hands if it's the first time that you hear about the MIQE guidelines, or if you heard of them before. Anyone? Okay, I mean, before Googling it, when you saw the title of the webinar. Because unfortunately, I have the feeling that they are quite neglected. But if you know them already, good, better. And if you heard of them, have you ever tried to implement them in your lab routine? I don't know, I cannot really see the show of hands, but I'm not understanding if you are with me or if you never did that, or it's okay. I'll assume you never did. Eventually, you'll stop me. Okay, so for the ones that are not familiar with them, or simply you need a refresh, MIQE is an acronym that stands for Minimum Information for Publication of Quantitative Real-Time PCR Experiment. It's quite a mouthful, right? And, oops, it's not going on. Okay, here it is. What is quantitative real-time PCR, often referred as qPCR, to be brief? It's a polymerase chain reaction that is coupled with a mean of quantifying the forming amplicons by using fluorescent dyes and standards, so that the buildup of amplicons at each reaction is revealed by changes in the fluorescence signal. It can be increasing fluorescence if you're using cybergreen. It can be a quenching if you're using Tachman. And then they are traced back to the initial copy number of a given sequence of nucleic acids, can be DNA or cDNA, for instance. And according to the type of references that are used, the results can be simply expressed by fold changes compared to a control samples, or actual copy numbers of the sequence of interest. Imagine when you were getting the virological test for the SARS-CoV-2, this would have the copy numbers of your infection, okay? So with this talk, I hope that I'll be able to convey why MIQE guidelines are important in warranting the reproducibility of qPCR experiments, that they constitute a framework that enable the scientists to correctly plan qPCR experiments, also by identifying the possible shortcomings. And finally, to critically evaluate the outcome of qPCR experiments according to how they've been performed, in case you're looking at your own experiments, the experiments run in your lab, or according to how they are described if you are reading a paper or a report. Okay, so before digging into the guidelines themselves, let's gather some general points about qPCR that might seem trivial, but allow us to better understand why this technique is particularly vulnerable to poor reproducibility. So first of all, the applications are really countless. It is an invaluable research tool, meaning that everybody basically uses it. And so, for instance, it allows for gene expression studies when it's coupled to the reverse transcription, or we can detect genetic polymorphisms, and it's cheaper in this way, for this application compared to deep sequencing, which can also give us similar information, but it's at a higher cost. It has also countless diagnostic applications for pathogens quantification, as we said, for the coronavirus, the determination of gene dosage, and the risk assessment for developing cancer or having recurrency of a cancer. And again, even if sequencing is getting in, PCR is certainly more affordable in the sense of the cost. So in the view of One Health that we cannot neglect, especially today, food safety is another important application for identifying contaminants. For instance, trace the food chain and also identify transgenes in case they are an issue with the regulation. For instance, in Europe, they are mainly forbidden. So transgene can be identified as long as they are not maybe inserted with CRISPR-Cas9, but this is another type of talk, of course. And last but not least, there are forensic applications, as many crime fictions, but unfortunately also reality taught us. Okay, so there are certainly many advantages of qPCR compared to other techniques. Simply the amplification, the fact that there is an amplification increases considerably the sensitivity of these techniques compared to the other. Then it is very flexible because it's sufficient to know the sequence of interest, to design the primers, and for the cost of a few euros, they can be delivered overnight. So it makes it also fast, not just flexible and rather cheap. Also, within the same sample, we can test multiple targets, and it became very user-friendly since it's kit-based. So it means that it doesn't need a strong biochemistry background to follow a kit protocol. Okay. But all these great advantages may quickly turn into disadvantages if the procedure is overlooked or approached with superficiality, with the results that often reproducibility is at stakes. It becomes an issue, and there is high variability intra-assay and inter-assay, and the lack of transparency and so forth. And the lack of transparency, again, many times it's unaware, like Valentina was saying before, for the manipulation of the images. It's just we don't know what we should report and what we should look at, so we don't, okay? So especially to me, it became an issue. I think it's quite let's say, shared this opinion that publications more often than not do not provide sufficient experimental details. And this is not just for qPCR, it's really a general problem, but somehow it seems to impact qPCR more than other techniques. And most likely, or at least this is my feeling, when you introduce a small bias or a small errors, and then you have such a big amplification step, the bias or the error gets amplified at each cycle. Okay, so consequently, without going into much details about the research versus the diagnostic application of qPCR, the need for standardization of qPCR for diagnostic purposes was soon recognized and implemented, because the accuracy of the diagnosis depends on the sensitivity and the specificity of the technique. So an assay that intrinsically returns false positive and false negatives with high rates is not just useless, it's harmful. And I think we all agree with that. We will not stick to a diagnostic assay that is like that. And also, I think we all agree that if I test in a lab or in another lab in different parts of the world, the results should be the same. So reproducibility between diagnostic laboratories is not even a question. It has to be reproducible. So why then we cope with the lack of reproducibility when it comes to research outcomes. And I know I've been provocative here, but just adding momentum to the next slide, because finally in 2009, it's when MIQE guidelines came out to the rescue in their, let's say, opinion, because I think we are not there yet. I mean, I don't want to be misunderstood. The guidelines are wonderful. It's just that we don't use them as much. So what happened was in the early 2000s, a group of scientists were worried, and I think it's really worth quoting what they wrote, that a real danger of the scientific literature being corrupted with a multitude of publications reporting inadequate and conflicting results, set out to develop and publish these guidelines. The aim was to provide the authors, the reviewers, and the editors with specifications for the minimum information that must be reported for qPCR experiment to ensure the relevance, the accuracy, the correct interpretation, and the repeatability. The MIQE guidelines were developed in the form of a checklist that we'll see later, and the principle that were followed were the MIBBI principle, that is the minimum information for biological and biomedical investigation, that are also applied to other techniques. So all of this has the main aim to ensure the integrity of the scientific literature, of the scientific process, promote, of course, reproducibility, and increase the transparency. Hence, this means that journals can and should refuse publication on the ground of non-compliance with MIQE guidelines, but I don't see that happening as much. So as reviewers also, we should be maybe, or anyone that is an associate editor should maybe be more attentive to this point. So just a brief timeline again of the history of the MIQE guidelines. In response to the retraction of several papers in the early 2000s, one were more notable, let's say, or more important, more impactful than other. One, for instance, was about retrieving or amplifying measles virus from autistic kids, and this was had a big impact on the public, for instance. And again, this breakthrough of the year 2005, published in Science, was then retracted in 2007. And these papers were retracted because they could not be reproduced anymore, not even from the original authors. So the decision from this group of scientists to write the first draft of the MIQE guidelines, and which is published by Bastin and collaborators in clinical chemistry. I think the second name is Vladimir Vance, which is, I mean, I think he still is, but he used to be the head of the genomic core of Embol at Heidelberg. So very, important name in the molecular biology field. And then they were followed by a series of updates on digital PCR, on single cell PCR. The last one was published in 2020 for the COVID. I think this is the last one. And then from time to time, they write commentaries and so on. So one in 2007 that was, I think, to the point, to what we are saying today, that is talking the talk. So we say that there are the MIQE guidelines and how we should use it, but not walking the walk, but we still don't do it. Okay. So just briefly, it's very small because I want you to go and open the actual paper later on when you have time, if you never did it before. This is the MIQE checklist, and it comprises 85 items, among which 57 are deemed essential, and they are the ones tagged with an E, if you can see that here, the little E, and other 28 that are tagged with a D that are called desirables. So if you have them, report them. If you don't have them, it's okay. But the essential, it's like you must have them in order to be able to publish your qPCR data. So such items are organized according to 9 categories, where we have the experimental design, the sample, the nucleic acid extraction, the reverse transcription, the target information, the oligonucleotides, the protocol, the validation, and last but not least, the data analysis, because even with the same type of data, according to the type of data analysis or quantification method that is used, or reference genes that are used, and how these reference genes are, let's say, stable or unstable, and how many they are, the final outcome can be different. So although this list has been designed to assist authors, reviewers, and editors, as you see with the arrow pointed to the title of the table, I find it extremely complete resource if you are approaching qPCR as a newbie, or after a while, or if you are in search of suggestions to improve the procedures in your lab, especially in terms of repeatability, if you are worried that this might be a risk. So just in case someone is not familiar with the procedure and nomenclature, here I reported a little a link to a video that you might want to go and see, but it's the same that I put also in the chat. And this will just walk you through the main steps, and which are the sampling, the nucleic acid isolation, the reverse transcription if you are interested in profiling the RNA, so you will have to transform it in cDNA. Otherwise, it goes directly from nucleic acid to amplification. So the amplification and detection are running in parallel. And this is what makes it real time, because I have the amplification, and then after the cycle of amplification, there is the detection, and it goes and it is repeated time after time, so that I can build a curve that is used then to go back and determine the initial, let's say, copy numbers or relative amount in my original sample. Okay. What I think it's very important to state is that there are so many ways that each of these steps can be done, and to know which one is the best or the most fitting, the most appropriate to our samples, we might have to test some of them. So each of these steps needs accurate planning, preliminary testing, testing of the condition, testing of the housekeeping genes, I mean the reference genes, which is the preferred terminology for the MIQE guidelines, in a very thorough way. Also checking how your primers perform. If they have poor performances, if they have too good performances, they are maybe recognizing also something else. So before doing the actual experiment, there is, I would say, at least 4 or 5 preliminary experiments to be done. Then once you set up the condition, they should be very repeatable, right? At least for this type of samples and this type of genes. But if you insert other genes or other set of primers, again, they have to be tested. So after this reminder of carefully running your preliminary experiments, because they need to be reported in the MIQE guidelines, let's brush up on some nomenclature here. So the preferred nomenclature is quantitative real-time PCR that can be translated to qPCR, but be careful to not call it RT-PCR, because this would be reverse transcription PCR, and it's an endpoint, not a real-time PCR. If I want to say that it's a reverse transcription quantitative, then it will have to be RT-qPCR, so reverse transcription quantitative PCR, real time. As I've mentioned already, the genes used for normalization, it's preferable not to call them housekeeping genes, but reference genes. And they can be external references that are added. They can be internal references as long as we measure their stability. And they should be more than one, usually. The probes that I've called Tachman probes in the guidelines, you will find them as hydrolysis probes. The term quantification should be preferred to quantitation, and the fractional PCR cycle that is used for quantification should be called CQ and not threshold cycles, as in many cases. Instead, we hear that it's usually the CT. So remember, the CT and the CQ are eventually the same thing, but it should be called CQ. Other couple of definition that I would like to share with you is repeatability and reproducibility. So the repeatability is a short-term precision or an intra-assay variance. So it measures the precision and the robustness of the assay with the same samples repeatedly analyzed in the same assay, the technical replicates. So you know that when you do qPCR, if you ever did that, you always put your samples with the same mixture of primers pairs in a triplicate, okay? And this is an accuracy of your pipetting and your mixing of the samples, and it's called repeatability, meaning that the three technical replicates should be sort of consistent. Otherwise, there is something sketchy going on in your preparation. Instead, what we are tackling, I mean, this is easier to see. You look at your data, you see the numbers. If they are not consistent, maybe you should arrange to, I don't know, maybe calibrate the pipettes or vortex and spin down your samples when you prepare them before aliquoting in the microwell. Instead, what we like to address with the MIQE guidelines is the reproducibility, so the long-term precision or interassay variance. And this refers to the variation in the results between runs. So I do an experiment this week and one the next week, or even between laboratories. I do an experiment here in Milan and you do an experiment in Tartu University, and we should more or less have the same results if the assay is reproducible. Okay, so what makes it reproducible is following two very strict, let's say, points and repeat them in the same way. So once I decided, because I did my preliminary testing, that this is the way that I'm going to collect my sample, process my sample, store my sample, do the extraction, the quantification, the quality control, the reverse transcription, and so on and so forth. I mean, the qPCR itself, the analysis, I stick to that. And if there are any changes that are introduced, then I will have to repeat it. I cannot compare. I mean, of course, I can compare to see what's different, but I cannot put them together and analyze as a whole has different biological replicates, because they are not actually. So the sample acquisition, I'm giving a couple of let's say examples here. We are not going to go through all the checklists. Don't be scared, because it wouldn't have any sense. It's really dependent on your type of samples. But if you have questions, we can discuss them. I'll give you just a couple of examples. So the sample acquisition constitutes the first potential source of the experimental variability. And this is my bias, to me, especially for experiments that target RNA, because RNA is much more sensitive to degradation compared to DNA, that it's inherently very, very stable. And I mean, traces of DNA that have been around for ages. Maybe they can be, have troubles with integrity. I am thinking mainly of forensics here. But generally speaking, in the lab, in the research lab, we don't have issue with the integrity or the stability of DNA. And this is because there are a lot of RNases, so enzymes that hydrolyze the RNA, kills it. And they are on our skin. They are immunological defenses for us against viruses. And they are really pleiotropic, they are everywhere. And so this is why you should be very clean, always wearing gloves. I like to use the double glove technique, because if I realize that I contaminated because, I don't know, I scratch myself or I touch something that I shouldn't have been touching, and I remove the glove, then my hand is wet, and I cannot put a clean glove right away. And so this leads my samples to be around for very long. So I usually use the double glove so that if I take out one, I can easily put another one back on. The other thing that we have to avoid RNases is that all the RNA procedures are done in a very confined, very controlled lab, which is super clean, and all the pipettes, tips, reagents, water, everything that you can think of, it's only for RNA, and it's appropriately cleaned. Only the PVC water and all of this. So if you want to know more about RNA integrities, I can go on for quite long, but I'll try to stop myself. So, but in any case, we should, independent of this RNA or DNA that we are looking at, we should always describe how the sample was obtained, how it was stored, if it was processed immediately or not, and if it wasn't processed immediately, how it was preserved and for how long. And also the cell type, and it's a mixture, it's a cell line, it's a sample of a tissue, because all of these make a massive difference on the type of results and the repeatability. So here are two quick samples. I took them from published papers. I did not disclose what they are, because really it doesn't matter what you do, but it's how it is described that I would like to go through with you. So let's read them briefly together. One is on ovarian tissues and the other one is on blood. So pair of ovaries were collected from the same animal and placed in a polyethylene bag on ice and transported to the laboratory within 1 hour after collection. The 3 largest follicles were selected from each animal using a ruler to measure follicle diameter. Follicular contents were aspirated using a 20-gauge needle and a syringe, and the follicle was flushed 3 times with glucose phosphate buffer saline. The cumulus oocyte complexes were identified by stereomicroscopy and separated from the follicular aspirate, and the remainder of the follicular aspirate and the saline flush was centrifuged to collect anthragranulosa cells. The collapsed follicles were then opened in half using a scalpel blade. The inside of the follicular wall was scraped with a microbiology culture loop to remove the mural layer of granulosa cells, which were then placed together with the anthragranulosa cells. The granulosa sample of each follicle was placed in an Eppendorf tube, sterile, RNase and DNase free, and plunged into liquid nitrogen and stored at -80 for quantitative real-time retrotranscription, PCR analysis. So the sample handling for the example 2, we said it's blood. So blood samples were collected from the purple crown fairy wren at the wildlife sanctuary managed by the Australian Wildlife Conservancy. After puncturing the brachial vein using a 25-gauge needle, approximately 70 microliters of blood was taken into heparinized capillary tubes and centrifuged at almost 14,000 G. approximately 10 microliter of compacted red blood cells, excluding white blood cells, was then added to a storage medium for different comparisons. So let's check these, let's say, description in the light of the checklist of the MIQE guidelines for the part that says sample. So if we refer to sample, we should have the description. It's mandatory. The volume mass of sample processed, it's desirable but not necessary. Micro dissection or macro dissection, processing procedures, if frozen, how and how quickly, if fixed, with what and how quickly, sample storage condition and duration. So I think that for most of the mandatory items, both description have most of the required let's say value or the required items. So we have the description of the type samples. For both of them, the type of dissection is very well described, especially in the first one. The processing procedure is quite well described. If frozen, how and how quickly? Well, this is lacking in example 2, if you notice, because it says storage medium, but it doesn't say how was the storage was conducted, if it was 4 degrees, if for -20, -80, liquid nitrogen. Instead, in the first example, it's more detailed. So it says plunged into liquid nitrogen, okay? So it's how quickly, it's immediate. As soon as I'm done with the isolation, they are immediately, it's not frozen, right? Then the storage is conducted at -80, so this is another type of indication for the sample storage condition, but both cases fails to report the duration. And even though -80 is pretty stable type of environment, it is reported that if RNases are present, they can continue slightly bit working even at very, very low temperature. And the amount of RNases in the case can depend on the contamination, as we highlighted before. And when we make the samples, it's not always the case that we can control the contamination, because if I go in the field taking a sample from an animal, for example, that has been that was naturally dead in the wildlife, and I want to take a sample from there, then I don't have much control on the contamination from RNases in there. But if I do it in the lab, then I can be very, very strict. And but again, it also depends on the amount of RNases that the sample has itself, because they are produced by the cells. So again, according to the type of cells and according to the type of tissue, the RNases that keep being in the samples even at -80, it's very variable. So this should be reported, because according to the sample type, this can be an issue or not, or a medium type of issue, let's say. So this was kind of easy to understand and go through. The sample checklist is, I think, the shortest, so it was the easiest to tackle together. But for the next step, where we have the nucleic acid extraction, and it's just the last example, set of examples that I'm gonna make with you, so that you have a framework, and then you can go check for all the other lists that are in the MIQE guidelines, and adjust them to your samples, to your practice in the lab, and so on. Okay, so going back to the nucleic acid extraction, these are the items that need to be reported or that it's desirable that they are reported. So the procedure and instrumentation used for the extraction with the name of the kit and details if modifications are applied compared to what the kit prescribes. Desirable, the source of additional reagents when they are used. It is very important to detail how and when the DNAs or RNAs treatments were done. So for instance, going back to the reverse transcription, PCR, it is fundamental to kill, eliminate all the DNA that is in there. Otherwise, when I amplify my cDNA, if there is a remaining contamination of DNA, this will also be amplified and eventually screw the assay. Okay, so it is very important not just that I say how it is done, but also that I check back that the contamination from DNA has been eliminated. And how do I do that? I have an extra, let's say, sample for the reverse transcription step, where the enzyme, the retrotranscriptase, is not added. So if there is DNA amplified from that reaction, it means it wasn't RNA that was reverse transcribed into cDNA, but it's a DNA contamination. Another way to keep double checking, excuse me, for DNA contamination is to have the primers that are designed in intron-spanning regions. So for instance, forward primers that is in one exon and reverse primers that is in another exon. If I have a residual contamination of DNA, the transcript, the amplicon that is formed is much longer because it also has the intron inside. This means that at the end of my qPCR protocol, I analyze the melting curve. The melting curve will show that there are amplicons of different size. Then I can take the endpoint results and run an electrophoretic gel, and I should see bands of different size. So this is another way to check it, that it's not reported here because it's the nucleic acid extraction list, but that we should consider. Another way, always in the primer designing strategy, you can have one of the primers that that is designed on a sequence that is intron spanning itself. So in that case, if contamination of DNA is present and the two exons are separated by the intron, the primer will not anneal. So you will have no amplification. Okay. So if I say things that are odd or you disagree or are confused, just stop me, okay? I cannot see your faces, so I don't know how I'm going there. Okay, I think I had a slide on contamination assessment and so on, but we dealt with that by now. It's very important to quantify the nucleic acid, and so the instrument and the method that are used to quantify should be reported, and these, I'm sure I have slides, we'll talk about it in a bit. The purity, conversely to what someone might expect, purity is only desirable, so it doesn't need to be reported. So the purity is usually measured by a spectrometer, a nanodrop, and it's given as the ratio of the absorbance at 260 to 80, and for RNA, ratio that is 1.8/2 is considered acceptable purity. I mean, a good purity, meaning there are no phenols residual or other contaminants. But if you don't have another drop, you cannot, or a spectrophotometer, you cannot really address that. Plus, there are other ways to check for the presence of inhibitors, and this inhibition testing, which is instead required, is essential. Also, the yield, surprisingly to me, it's only desirable, because it's true you have to say how much nucleic acids you put in each reaction, and this is of course mandatory for the list of the PCR part, but you don't have to necessarily say how much yield you had from each sample. So this is discretionary, you can say it or not, but if you have it, why not? I mean, to me, I like to show that the yield was good when I have, especially when I have experiments that or results that are not super clear cut, so that I know that the yield or the purity or the integrity was not the issue, that there is something else in there that I should go look for. Because if I was a reviewer and someone would give me something that it's hard to interpret, the first thing that I would say, it's like, oh, okay, show me your bioanalyzer or your, let's say, quantification and purity and so on, because I'm not sure that your RNA preparation is good. So RNA integrity, we'll look at that in a minute. Okay, so we can proceed maybe. I think that I was carried in the moment, and I already anticipated some of the things that are in these slides, but I'll go through them in any case. So if you have a question, you can ask. So the extraction method have a substantial effect on both the RNA yield and the RNA quality. And some of the factors that influence extraction efficiency are, but not limited to, the homogenization of the samples, the lysis of the samples. So if the lysis buffer that I'm using or the mechanical lysis that I'm using is not good enough for a certain type of sample, then of course I'm not extracting all the RNA or the DNA that I'm supposed to have. So when you have culture cells or small pieces of tissues, it's easier to homogenize and lyse. But if you have, I don't know, bones or things like that, or even microbes, I mean, bacteria are very hard to extract sometimes. The density of my target, and also the physiological status. Am I collecting, again, from an animal that was dead since a couple of days in the field, and someone called me and I'm going to the wildlife park to get my samples, or it's a cell culture that I'm retrieving in the lab? So this makes massive difference on the purity, the concentration, and the integrity of my RNA, the expected ones, right? So all of these details must be provided, so that eventually, if we can control the conditions, then we will try to keep them always the same. And I would like to talk to you about some RNA extraction kits that select specific RNA species, and this might introduce a bias toward one RNA type. So for instance, if you're using Dynabeads for the extraction of your RNA, this is how Dynabeads work. So they are coded bits with long strings of T's, so that they preferentially bind to the poly-A tail of the transcript. So this means that what are the transcripts that have the poly-A tail? They are the messenger RNA. So all the other type of RNA, the transfer, the micro, the small nucleolar, the non-coding, the long non-coding, all of this goes to waste, okay? So you have to understand if you're interested in them, or if the paper that you're reading wants to look at them and uses Dynabeads, maybe there is something that it's not totally matching. And another word of caution, but this really depends, I think, on your sample. I'm very sensitive to that because we work with oocyte and embryos, and this is an issue for this type of samples. the same RNA can exist during the lifespan of an oocyte or an early stage embryo with different length of poly-A. So if I use Dynabits or also in the reverse transcription, some kind of compounds that preferentially binds to long strings of A, I might be introducing a bias towards the transcript that have a longer poly-A tail compared to the other. And I actually saw that in my experiments. And when we realized that, we used it as a comparative assay that worked well for mouse oocytes. We were having a striking difference if using for the reverse transcription, in this case, oligo-DT, so something that would preferentially bind and select the poly-A tail compared to random hexamers. With other species, might be not exactly like that. So for the bovine, for instance, there is a certain length of poly-A tail that already allows for a good, let's say, affinity for oligo-DT, even when the poly-A is not super, super long. So the bias was not there so much. But again, this is something that you might have to check with your own samples. For instance, it's becoming more and more, let's say, evident that there are additional steps of post-transcriptional regulation that can influence the poly-A tail, not just in oocytes and embryos, but also in other cell types. I mean, neurons comes to mind. And so if you're if you are working in neurosciences, maybe this is something that you want to consider, the type of extraction kit that you're using and the type of reverse transcription methodology or priming that you're using. And actually, this is, I mean, the Dynabits can be well understood, right? There is an intrinsic bias, it's there for purpose, and it can be used with purpose. But unfortunately, during my experience with RNA extraction, we also observed that we have differences using different kits that are supposed to work more or less in the same way. So again, be very careful of what you do, and maybe try to, if you see something that is different from what was reported in the literature, maybe you might have to consider to change the extraction kit. This is what really happened to us. And again, to solve this issue, I think transparency was key, because we were seeing something that was not expected and were getting freaked out. So we had a very honest talk, me and my PhD student, with her doctoral study panel that has a big expert in RNA extraction. I don't know if you know Marc-André Sirat. So we were talking about that with him, and he was the one telling us, try to change the kit, the brand of the kit. And it actually reversed the things back. So before freaking out, maybe ask a colleague and be transparent about that.
Sorry, a second. I don't know if you can see it, but there is a question in the chat.
I cannot see it. Sorry, but
so I can read it.
Open the mic. Yes.
Yeah, I can read it for you.
Thank you. Yeah.
So reading the nucleic acid extraction quality, what if the sample is hard to work with and the amount yield is also small? How to implement the guidelines then? As it is not possible to do all pre-tests, and also what if the nanodrop and qubit yield values are totally different? Which values is then the preferred and to proceed with downstream applications?
Okay, can I take it step by step? So I'll try to answer, and then if there is something that I'm missing, let's see if I can help you in there. Yes, when there is issue of samples, then you will have to deal with what you have. If it's very hard to retrieve and you you have to be open about that, transparent, report everything. So these are good guidelines to, let's say, plan your experiments ahead. And if you have enough samples, do everything. But if your limitation is an extreme sample restriction, you can use the guidelines to have, you know, best practices, and then you exactly report what you did, because the issue here is being reproducible. So if someone else or you yourself wants to do the same, perform the same experiment again, you know that changing the conditions of the previous experiment might take you to different results. So I don't know if I answer the question. So I'm suggesting to use the MIQE guidelines to plan your experiments, right? But actually what they are for authors, reviewers, editors. So it's for publishing. So they say what is essential, but if you cannot make it with your samples, then I don't see very much way around it, except that you be super transparent on what you did. So this is my, and then for the nanodrop qubit question, I think I would go with qubit. It's, I mean, it's not just me, but it's reported that, I mean, nanodrop, it can also be an issue, you know, of pipetting or having the cell super clean, and it's not always the case. People are using it. I mean, it's shared with other labs. You never know what's happening. And I don't know. I trust more the qubit, but it's reported that the qubit is more accurate compared to, maybe not as sensitive, but I mean, you can work around the sensitivity if you include, you know, known standard of initial material. So this is the way we do. So for instance, if I make a preparation of lysate of oocytes, in order to be able to quantify them with the qubit, I would need too many. So what we do is we put a known concentration of external RNA. You can buy it from Promega. You can do it yourself with an in vitro transcription kit if you want. And then on top, you add your sample. And then by subtraction, you should be able to adjust the thing. So we are lucky enough that we have a qubit in our lab and we have a nanodrop in the premises, in the facility. So eventually I can run both all the times. My colleagues are very nice and allows us to use it when we need it. So if I'm in a hurry and I know that I have a lot of cells, a lot of RNA, I don't lose time in doing the qubit maybe. And also, I mean, the quantification, it can also be done just by counting the cells, actually. And this is something that I've stumbled upon recently. So the way we are used to think of quantitative PCR, but also big omics, is that we need to start from the same RNA amount. But it has been reported from a super nice lab that it's now at Toronto University, and is Miguel Ramalos Santos, the PI. He worked in San Francisco before, and this is how I know him. It's like different cell types, they can have different basal level of transcription without having differences in the type of transcripts, but just the total amount of transcripts. So if you use the same input, you are going to miss this type of variability, which he calls hypertranscription, and it's typical of stem cells and of embryos, and that other cells that don't have. So maybe you can just count the number of cells if this is an issue. and then compare the amplification, the CQ that you have, normalized not by the initial amount in terms of nanograms, but in terms of cell number. And for the oocyte, it's like that as well. We use oocyte equivalents, for instance, and not concentration of RNA in many applications, because there is a wave of RNA degradation during a certain stage of oocyte development. So if you compare two different stages and you are quantifying the RNA, you are putting more oocytes in one treatment compared to the other. So we use oocyte equivalents so that we can see this RNA degradation, because we know that it occurs. Okay, so I think I derailed a little bit. I don't know where I was anymore. Were we here? Okay, so I think we went through the DNase, RNase treatment and the contamination assessment already. So the nucleic acid quantification here, did I answer the whole question, or there was something else that I missed?
I mean, there's no reply, so I think it's okay. You replied everything.
Okay, thank you. I blabbed a lot, sorry. So I'll try to come to the end because we're out of time. So basically, we also talked about RNA integrity. So we have to show that there is no degradation, or if the degradation is there, we should say it, right? Because in some fields condition or cell types, it's really impossible not to have degradation maybe. So the degradation should be something that comes from the sample, of course, and not from the handling. But we should provide the RNA integrity in order to be able to correctly interpret the data. So the RNA integrity can be done with RNA gel, but of course this requires a lot of RNA, and we should see two sharp bends on the 28S and 18S ribosomal units, so the ribosomal RNA, which is okay, but still it might be that the ribosomal RNA, being very much abundant, is not as much sensitive to degradation as messenger RNAs or other type of RNAs. We can do a gel electrophoresis microfluidic base, which would be the bioanalyzer. The type of interpretation is the same with the 20S, 28S, 18S bands. So it might still have this little bias, but it's usually very clear cut. But if I don't have much RNA, so I cannot do an RNA gel, I don't have a bioanalyzer available, then the suggestion is to perform a 3', 5' integrity assay. How does it work? It means that after the reverse transcriptase, before doing my qPCR, I do an additional, let's say, preliminary PCR to check my integrity. And this means that I have to design primers that are very far, one at the 5' and one at the 3' of my mRNA. And if the RNA is good quality, the integrity is high, the two primers will anneal and amplify, and so you will have the band of the expected size. But if the DNA is degraded, they won't be able to give any amplification. So this is also accepted by journals, and actually it was what Vladimir Bent was suggesting me, where we didn't have the access to the bioanalyzer. Okay, so these are two graphs that comes out of the bioanalyzer. This one represents the peaks of the 18S and 28S, which are here and here. And this is a representative graph of a good integrity sample. And here instead, you can see the degradation. So the peaks are less sharp, they are lower, and also we have all these RNA species in here. These ones might be small non-coding and micro RNA and so on. But again, this really depends on the type of samples. Okay, so my final remarks, mainly, I mean, these slides on qPCR, I would like to remind you that most qPCR data are not absolute, but relative, unless you have copy numbers. And the standardization makes it all. The reference genes makes it all. So even if you have very low amount of samples, also include multiple reference genes. This is something I would not compromise on. Maybe you can use external references if you don't want to increase the amount of RNA that you use for internal references, but this is mandatory. Actually, this is what we did with the Corso site when we did single site PCR. We were using an external reference. We also compared it to the internal just in case in some preliminary experiments to see if they could give the same type of information, and they did. So we elected to use one against the other. Again, the detection of the presence of the messenger RNA provides no information on whether that mRNA will be translated into a functional protein. So be very careful about that, also depending on the type of samples. I'm going back to oocytes, to embryos, to neurons. If there is the mRNA, it's not said that this is right now translated into a functional protein. So the take-home message here is always couple your qPCR experiments with some other techniques, immunohistochemistry, Western blotting, other protein quantification methods, or functional type of data. They should all, let's say, be together in order to allow for a correct or interpretation. And then the final remarks, but I think about research integrity, but I think Valentina already talked quite a bit about them. And I think that after all this list of guidelines and why repeatability is important and so on, it's quite obvious at this point that correctly performing and reporting qPCR experiments is part of the research integrity, because we cannot say things that are not replicable. We cannot afford it for our labs, for the trust that the public has on us, and so forth. So if you look into the checklist of these 5 principles of research integrity, among the rigor, honesty, transparency, accountability, you will find everything that we've talked about today, but maybe a little bit seems a little bit out of the qPCR experiment, the part of the respect. But I think we should really put it in place in the sense of respecting the data. So trusting the data. If something is off, there should be an explanation for that. Trust the people that perform the experiments, in the sense, not blindly trust, but give them the respect that they need, because a student or a technician that feels free to go to the tutor, the mentor, the PI, the colleague, and say exactly what happened, it's a person that will be transparent. They don't have to cover. If we create an environment that is a sick environment in the lab, people will hide things from us. So this is something that we should really consider in our daily lives. And this then goes back to the replication crisis. So these graphs are taken from a publication in Nature back in 2016, and it's striking to me that 50% of the scientists that were interviewed said, yes, there is a significant crisis. And some said a slight crisis, making up for overall 90% of the scientists saying that there is somewhat a crisis. but only a minority of them, or I mean 65%, took action in order to reduce the crisis. There are still 35% of the same scientists that acknowledge that there is a crisis but didn't do anything. So be proactive, at least for qPCR. My MIQE guidelines are there, and they are there to help. Okay, and if we want to stop the trust crisis that the public has in the respect of science, we should really look into our houses first and try to stop the replication crisis. Okay, and here are some of the references that might be helpful to you. And last but not least, I would like to thank you all for sharing this time with me and to acknowledge the members of my lab and the fundings that allowed me to be here and perform experiments for all these years. I think I'm done, yes.
Thank you very much, Federica, for sharing with us your knowledge and experience. I think it was also very insightful and was a good learning experience for all participants. So now is the time to ask more questions, as one we had in the chat. Do we have any other? Yes, please, Kasun, go ahead.
Hi. Can you hear me?
Yes.
Yeah, very nice presentation, the MIQE, and how not a lot of people really follow it, and the papers really don't care and whatnot, right? So I usually do quite a lot of absolute quantification. In MIQE, there is nothing about absolute quantification as far as I know in 2009 one. Is there another document or like standard that we have to follow, or it's fine that we do absolute quantification and we follow just the parts that in MIQE that actually applies? What is your opinion?
No, I think so. I think that, I mean, it's very strict if you look at it and you want to be absolutely compliant. But I think a lot of good science comes out without all this information. The importance is being aware they should be there. And then if they cannot be there because of your sample, because of your type of procedures or so on, be transparent and say to the reviewers and say to in your publication. The important thing is to establish a way for things to be interpretable and be repeatable. So if I look at, say, qPCR plot, and I see, and there are no information like on the reference genes that were used, how the stability of the reference genes were established, then this is the first thing that I'm going to ask to as a reviewer, right? But if you do absolute quantification, then you don't need the reference genes, so you shouldn't be compliant with that part, right?
If I can jump in, if this can be of any help. Long time ago, when all this story started, there was a very nice, tech note or something from Biorad that did a clear that distinguished between how to deal with MIQE guidelines for quantitative or absolute. So it's there.
If you open the paper, you should, especially the last versions, they should be there. I mean, they don't tell you have to do it if it's not pertinent to you. You have to do it if you use reference genes, of course. But if you don't use it, I mean, have I understood correctly that you use that you give copy numbers?
Yeah, we work with extracellular vesicle-based RNA, so there's no reference genes to be had. Yeah, so as well.
So you need to find. I think there might be something on the extra vesicles too. You should find the application of MIQE guidelines to the extra vesicles.
So when I look online for these minimum requirements, the paper that comes up is the 2009 version. is there a place that I can get like the later, like editions, later updates? Because the thing that comes up and mostly referenced is this 2009 version, and there's nothing about Absolute there.
Yes, I think you should be able to find it. And if you don't find it, you can write to the MIQE group. They are still active. But I can try have a look for you also and see what I find.
No, it's fine. Thank you very much.
Thank you. And we have one more question in the chat. So the question is, in the guidelines, you see.
Yes, now I see. Sorry. In the guidelines, it was that we should use CQ values instead of CT values. Does this somehow influence the data? No, it's just a nomenclature. So CT is something that is used by a brand. So if you look at the guidelines, they don't name brand ever. So they say microfluidics RNA gel, not to say bioanalyzer. So since or hydrolysis probes, not to say TACMAN, because they don't want to be, having conflicts with brands. And it's the same issue here. CT was used by a company that made real-time machines. And so they say, let's use CQ because we don't want to be biased towards a company or another. Yeah, but if your machine says CT, if your machine says CT, it's good. It's just the same as CQ. Just when you report it in your paper, name it CQ.
Is that enough answer? I hope yes. So do we have any other questions?
Okay, I'm glad if I could help.
Yeah.
But if there are more things that you would like to ask, if they come up later when you read the guidelines or so on, we can get in touch. I'm not sure I'll be able to actually help you. I've told you my experience is sort of limited into the reverse transcription qPCR for very specific samples, but we can brainstorm together or find someone that can help us.
Yeah, definitely. As we are in the project together anyway, still by 1.5 of the year. So
I think people are always willing to share their knowledge, and at least we go to the ones that are willing to.
Yes. So thank you very much, Valentina and Federica, for very nice, very educational